Hi, this is Jessie Packard.
And Heather Benson.
And we're just going to go over the Lesson 12 group problems with you. So for question 1, it gives you a list of things that are involved with the LAC operon process and everything involved there. And so for part A, it just wants to know which of the terms describes an example of a regulatory protein. And that would be your activator, your repressor, and your CAP. And then for part B, it wants to know the terms that describe an example of a small effector molecule. Those are going to be your inducers, inhibitors, your allolactase, and your CAMP. And for part C, which terms describe an example of a sequence that's actually found on the DNA, and that's going to be your +1, your -10, your LAC-A, and your LAC-Z. And so we learned about the +1 and -10. Those are involved in transcription. So those are going to be for DNA. And then your LAC-A and LAC-Z are structural genes, also on DNA. And for part D, if we're talking about what is actually on the LAC operon itself, then the ones not on there are going to be your allolactase, your CAMP, inducers, inhibitors, and your activators. Those aren't actually on the operon itself. And then if we're talking about the process of the whole LAC operon, then it would be none of them because they're all involved in the process. And then finally for part E, we kind of already covered this, but it just wants to know the structural genes, and that's going to be your LAC-A and your LAC-Z.
Okay, awesome. So I'm going to talk about number 2. And so is that showing okay?
Yep, that looks good.
Okay, so for number 2, it just has a lot of different fill-in-the-blank questions. So for part A, the LAC operon is regulated by 2 inducers, allolactose and CAMP. Part B, the binding site for sigma factor and RNA polymerase is LAC-P, while the cap site is the binding site for the activator that regulates the LAC operon. Part C, the enzyme adenyl cyclase converts ATP into CAMP. We learn that in the notes. Part D, the -35, -10, and +1 sequences are found within the LAC-P portion of the LAC operon. Part E, the LAC-I gene encodes the LAC repressor, while the LAC-Y gene encodes lactose permease. Part F, attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And part G, an E. coli strain that contains 2 copies of the LAC operon is called a merozygote strain.
Awesome. For question number 3, it gives you different cultures and their conditions, and it wants you to figure out which of the strains are going to turn blue because of the XGAL factor. And so for the solution to turn blue, you need both the IPTG and the XGAL factor to be present. So keeping that in mind, for part A, your IPTG is absent, so that's not going to react with your XGAL and it's not going to be blue. And then for part B, you have both your IPTG and your XGAL factors present, and because glucose is also absent, then that allows your solution to turn blue. And for part C, your IPTG is present this time, except your XGAL is absent, so it's not going to be blue. And for part D, your IPTG factor is there. Your XGAL is also present, but because glucose is present, it's not going to allow those two to metabolize, so it's not going to be blue. And finally, for part E, you're missing your XGAL factor, so it's not going to be blue as well.
So for part number 4, they're talking about several different E. coli strains, and they give us a bunch of different important things that we need to know. So it shows what the defective gene and the wild type gene will be indicated as, and the defective operator sequence. And it tells us that IS produces lac repressor that can bind to the operator but can't bind to allolactase, and that glucose levels are low. So those are important things that we need to know to be able to answer it. So for part A, it gives us this here. And so what we can tell from this is that everything's functioning correctly because we have the wild type genes. So LAC-I is functioning and things are binding to the operator correctly. And this is because all parts are wild type. And so it's able to bind to the operator, and the transcript isn't able to go forward, which means that it is inducible. For part B, what we learn here is that these are all wild type, but the IS is present. And it says, we go back to what it says, IS produces a lac repressor that can bind to the operator but cannot bind to allolactase. And so that means that it binds to the operator, but nothing can bind to the repressor to remove it. So that means that it will be non-inducible. For part C, we have this here, and we can see that we have wild type here, but this is defective, and here we have a defective operator sequence. And so we have this on the chromosome, and then over here on the plasmid, everything is wild type except for the defective operator sequence. And so this ends up being constitutive because there's a defective promoter and operator on this one, and a defective operator here. And this means that, so there's two operators going, and they're both functioning. So repressor proteins are being produced correctly, but there's a bad operator, so it ends up being constitutive. For part D, it ends up being non-inducible because on the chromosome we have all wild type, but on the plasmid we have wild type with the IS producing the lac repressor. And so we still have, like, things are still bound to the operator. And so, so as it says up there, the IS produces the repressor that can't bind to the operator but can bind to the allolactose, and so all of this makes it so it's non-inducible. And then for part E, we still have transcription happening twice because we have the chromosome and the plasmid. And what we have here is that it's constitutive because the IS, the repressor, can't bind to the allolactose, and there's a bad operator over here. And the lac repressor can't bind to the operator, so it's constitutive.
Perfect. And so for question 5, this is talking about the experiment that was performed to get all of these strains of E. coli, and it wants you to figure out how yellow your solution is going to be. And so 0 represents that there's not going to be any yellow in your solutions. 1 is like a medium amount when 1 copy at least of your lac operon is transcribed. And then 2 indicates that there's going to be a lot of yellow produced when there's 2 copies of the lac operon transcribed. So for part A, there's going to be 0 when there's lactose and 0 when there's not lactose present. And this is because of the negative on the I, and that's because there's no lac repressor. However, there's a negative on the P as well, which means there's not going to be any starting of transcription. So that's not going to produce any yellow at all. And for part B, it has a C on the O, meaning it has a bad operator. So this is going to keep the operator open and functioning all the time, whether there's lactose or no lactose. So you should have a 1 for each of those, for lactose and lactose. And for part C, the chromosome is perfectly fine, and it's going to function with lactose. And then for the F plasmid, the only thing wrong with it is it has a bad operator, so that's going to keep functioning all the time, meaning it's going to work with lactose and without lactose, whereas the chromosome part is only going to work with lactose. So you should have a 2 for lactose and a 1 for no lactose for part C. And for part D, your chromosome is going to function. It just doesn't have a repressor, which is fine. And so that is going to show up with lactose and without lactose because there's nothing repressing it, so transcription is always going to keep going. And then for your plasmid, there's no start again because your P site is defective, so there's not going to be any transcription happening there. And finally, for part E, your chromosome is pretty much perfectly fine. It's got a no repressor as well, so it would keep functioning normally. However, because of your plasmid that is always being repressed by the S on the I, it's going to kind of inhibit the chromosome from functioning, so you should have a 0 for both columns for part E, whether there's lactose or no lactose. So.
Great. Well, thanks for watching, everyone.